thank you all for attending and coming to my talk. I'm really excited to share this work that we've been doing on the white nose syndrome invasion dynamics in Texas. And just getting used to seeing everything pop up. Although it's not Hawaii, Texas has really been incredibly impressive in terms of its bat fauna and in terms of its caves, which I'm excited to share with you. All right, advanced slide. Okay, so white nose syndrome is an invasive fungal disease. And to talk a little bit about invasive diseases, we had the amphibian apocalypse, which was caused by chytrid fungus, and it's really just a sign of the times now more than ever how invasive diseases can rapidly wipe out species and cause unprecedented declines when new pathogens end up in new environments, which I'm sure we're all acutely aware of with the emergence of COVID-19. So white nose syndrome is a fungal disease of hibernating bats, so it affects them during the winter. And I'm just going to turn off that. Okay. So this has been happening since about 2006 when white nose was introduced to North America, presumably from somewhere in Europe or Asia, and has since caused devastating declines. All right, advance. All right, so what happens is this fungus invades the skin of hibernating bats during the winter. So here is a UV lit photo of a bat that is infected with white nose syndrome. So you can see that the fungus, Pseudogymnoascus destructans, glows or fluorescence under UV light. So you can see it's invading the wing tissue. Since it was first discovered in 2006 in one cave in upstate New York, you can see with the color progression how it has spread through the northeastern United States and up into Canada. And more recently, in the past few years, it has reached as far south as Texas. So we can see that was first detected or suspected in Texas in the 2017-2018 winter. And now we're starting to see reports of dead bats in Texas that have presumed to have died from white nose syndrome. So they've tested positive, and there have been more reports of them across the landscape. So what this, a little bit more background on white nose syndrome. So population declines when a cave is infected with white nose exceeds 90% for multiple species. So here we have a hibernating little brown bat, and you can see after a few years of having the disease, the population was at 20 and then closer to 5% of what its initial population size was. So this is substantial and some of the most dramatic mammalian declines we have ever recorded. And it's especially concerning because it's affecting a large number of species that are hibernating. All right. So what happens when the fungus invades the tissue is that these wing membranes are part of how bats breathe throughout the winter. So hibernating bats are in environments that have very high humidity levels, so often 95 to 99, 100% humidity in the cold air, which allows them to do some gas exchange, so breathe, over the skin tissue of their wings. But as the fungus invades this delicate wing tissue, what happens is bats are no longer able to exchange oxygen over their wings, and so to compensate for that, they have to increase how much they're respiring with their lungs. So hibernating bats actually breathe very infrequently. They will, every few minutes or so, rapidly exchange all the gas in their lungs, stop, and be kind of apneic and let that gas exchange happen. However, if their wings aren't doing as much of the heavy lifting for respiring, they have to do more of that breathing, which causes them to lose more of their water. So they start to dehydrate because they're increasing their pulmonary gas exchange. There's also dehydration that's happening as liquid seeps through the open sores in their wings. And overall, this change in hibernation physiology increases how much energy they spend throughout hibernation. So one thing that happens is that these bats warm up from hibernation more frequently. So hibernating bats are down a few degrees above the ambient temperature of the cave. So that would be about 10 degrees or 8 degrees Celsius. I'm sorry, I don't have the Fahrenheit in mind. So actually very, very cold. But then periodically, every few weeks, they will boost back up to normal mammalian body temperature, be active for about an hour. They might sleep, groom, respire, things like that, move around. But what happens with white nose syndrome is we see these frequencies of arousals. Thanks, Nate, for the temperature translations. So the frequencies of these arousals increases. And because these arousals are what bats spend 90% of their energy budget on over winter, even though they're very brief, this basically causes them to burn through all of their fat stores meant to survive the winter far too rapidly. And so in the north, this is where we see that dead, emaciated bats are found on the landscape in snowbanks as they leave caves in search for food. So that's the overall pathophysiology of what happens with white nose syndrome. All right. And then as a consequence of this, so we detect the fungus, Pseudogymnoascus destructans, about 2 years before we start to see the mass die-offs happen. So this graph here shows sites where PD was present in the northeastern United States. On the left y-axis, we have the colony size and the red line, and on the right side is fungal load. So higher levels mean higher intensity of fungal infection. What we see is after the fungus arrives, we have a couple years before the mass die-off happens, and in those couple years, PD loads on bats start to increase. So these are data for the little brown bat in the top 4 panels here, and Perimyos subflavus, the tricolored bat, which is a species. Oops, I clicked on something that changed my view. So just checking, can everyone, Nate, can you still hear me when I clicked my different thing and you can still?
You came back.
Okay, so I'm back, and is the presentation going?
No, it's not going.
Okay, I was gesturing and it's
here. I have a solution here. Just hold on 1 second. I did.
Okay. This is one of my first presentations on WebEx. It's not the PowerPoint. Okay. Oh, I see. I've got to go back to the share. PowerPoint. Okay.
Perfect.
Hey, all right, we're back. Thanks. All right. So where was I? Basically what we see is after a couple of years, after fungal invasion, fungus loads go up, and that is matched with when these bat populations decline. So now it's been a couple years since PD has been present in Texas. We haven't seen any declines yet, but this is about the time when we expect to start seeing mortality, and that has played out, at least in some of the first reports this winter. Okay, so the fungus stays in the hibernacula, and so it remains on the substrate of the walls, and then what happens is when the bat the next winter, they become reinfected with the fungus. So this is a cold-loving fungus that grows in these cool temperatures. So bats are able to clear the infection and do all right during the summer months. And it's when they return to these hibernacula that become reinfected. All right. So what we urgently need are scalable solutions to increase bat survival over the winter where this mortality has occurred, or where we're to prevent mortality when it's going to occur. But to do that, we need to have an idea of what sites are going to be most at risk and where this is happening. All right, so now to shift into what is going on in Texas in terms of this invasion. So there are a few things that are going to make the situation in Texas different. First, we have several species of bats that are not in that have not previously been exposed to PD, so we don't know how they will respond to white nose syndrome. Some bat species do very poorly, such as the little brown bat, the northern long-eared bat, Perimyos subflavus, the tricolored bat, but others seem to be fairly resilient. So we need to see how the bats in Texas will respond The other thing that you're probably quite aware of is that the winter duration is quite a bit smaller, and most of this mortality is happening in the last few months of winter. So there was some hope that bats wouldn't have the fungus long enough to show these signs of disease. And of course, the microclimates in these hibernacula are also different. They're warmer than a lot of the northern caves, which warmer being still at cool temperatures, still close to that 50 Fahrenheit mark, and still well within the growth range of the fungus. So Texas is pretty exciting in that it has 30 species of bats. That is the highest diversity in any American state or Canadian province. But we're going to focus right now on 6 species that commonly hibernate in Texas. So not all of these species are hibernators. Some of them are migratory, and some we just don't have good data for where large aggregations of them would occur in the winter. So here we have the tricolored bat, Perimyotis subflavus, and as mentioned in those earlier slides, they are quite susceptible to high mortality from white nose syndrome. So you can see this bat is covered in lots of little dewdrops, so they prefer hibernation environments that are incredibly humid, 100% humidity, and they also hibernate solitarily as opposed to in groups, and this is quite small, about 4 grams. So these are one of the ones we are #1 concerned about. The other major species we are interested in is cave myotis, Myotis velifer. So these are larger myotis species. They form large aggregations of several hundred bats in clusters, larger caves, and we don't really know how they're going to start responding to PD because the myotis species are quite varied in how susceptible they are. The Townsend's big-eared bat, Coronarinas townsendii, is thought to be a low white nose mortality risk based on parts of its other range, and because they prefer much drier hibernation environments and much more variable temperatures, those conditions seem to be less good for the fungal growth. So the big brown bat, as mentioned earlier, is less likely to have white nose syndrome related mortality. And again, this seems to be as part of their adaptation to drier hibernation environments. And when you think about those adaptations to drier environments, part of what that means is their resistance to water loss, which is what's driving some of this pathophysiology. And then finally, the Mexican free-tailed bat, Gerarda brasiliensis, is thought to be a low white nose mortality risk for similar reasons. And a large number of these species is not hibernating. Sorry, can you describe?
Sorry, Kristin, that was for Robert Trudeau asked a question about a particular slide you were on, and I don't know which slide that was, and it had something to do with population increase. So I was asking for more detail on that question so that at the end we can then ask. Sorry. So just ignore the chat window unless something drastic happens.
Okay, I'll ignore the chat window and look for you to yell at me. All right. Okay, and back to clicking. Okay, so this is the PD invasion front within Texas. So you can see that it first entered the state of Texas, northern panhandle area, and now more recently we have detected PD in central Texas. So that's why some of the early work was done in the panhandle, because it's closer to Oklahoma and some of this invasion front there. So it's been in Texas for a couple of years. And what we do to look for the fungus is we go into these hibernacula in early winter, around December and late winter, and we take swabs, and we're basically rolling them on the substrate and on the bats to get fungal DNA. So this is actually very similar to the nasal swabs people have been getting for COVID-19, where you ram that up your nose. We do not ram this up bats' noses. We roll it gently over their nose and gently up and down one wing, get fungal DNA, and then amplify that to see how much of that is there. So featured here on the right is Fran Hutchins, who is our lead for subterranean work on this project, and is incredibly familiar with these caves and the populations and what's been happening. And on the left, Melissa is one of our crew members who's assisting with swabbing bats and doing these counts and things. So that's basically what we're doing when we're entering. And then I want to talk about the two metrics I'm going to talk about in terms of what we're seeing in these patterns of fungal disease. So the first one is prevalence. So if we have 10 bats here and 3 of them are red, that's going to be 30% prevalence. So 3 of the bats are showing positive for having the fungus. So I'm going to present now some data from other species in North America. So we can see the first 3 species, Septentrionalis, the northern long-eared bat, Matus lucifugus, the little brown bat, and Pipistrellus subflavus, the tricolored bat, which we have in Texas. After, and on the x-axis is years since detection. So after about 2 years, you can see the prevalence ramps up to about 100%. So all individuals are testing very highly positive for this, whereas you see on the right, other species that are less affected by the disease, so big brown bats, Pseudogymnoascus fuscus, Myotis lebii, Myotis graciens, don't reach that higher level of prevalence. So it's less individuals come into contact with the fungus and grow it, even after several years, which it's been introduced. So these are patterns we want to see in our data. Are we having patterns like on the left with highly susceptible species or further on the right? All right. Oh, that did not show up perfectly, but that's all right. So the next is fungal load. So then if we take only those 3 bats that tested positive for the fungus, we want to know how much there is. So basically, do you have a little or do you have a lot? Because fungal load is associated with mortality. So higher loads are what is associated with death in these bats. And so on this graph, we show the load on the y-axis in attograms of PD and days of hibernation on the x-axis. So the overall pattern is that throughout winter, individuals will accumulate higher fungal loads. So that increases. Oh, that's where. And then as years go on, we can see that the species that do more poorly attain higher loads, and that levels off. So between 6 and 8 attograms of PD is what we are seeing in fungal loads of species that experience high mortality. Whereas if we see lower loads, closer to 5, those species tend to be doing better. So that's the patterns we're looking for in the data we see here. All right, so this then is where we would do a bat swab on his little nose, an under swab on the substrate very close to the bat to see if some of this fungus is shedding onto the substrate and how much. We take those under swabs because often those samples can help us detect really low levels of the fungus before we start detecting them on the bats. So that gives us kind of an early view. And then here is just a station. So we have some stationary points within the caves where we go to the same part of the wall year after year and see what's happening in that environmental reservoir with the fungus. All right, so these are our data for prevalence in North Texas from this past August, December, and February. So in the top, each of these panels is a different cave site. I've just blocked out the identifying information. So Myotis velifer is shown in red, and we can see between December and February, we had an increase in the prevalence of bats that had tested positive for PD. So we're seeing higher levels of prevalence both on the bats and underneath them in terms of the cave Myotis, and then as well in terms of the tricolored bat. So we have one cave here where tricolored bats are not testing positive yet, and another where they are and the loads are ramping up. So this means there's reasons to be concerned for both of these species, the Myotis velifer and the Perimyotis subflavus. The big brown we swabbed did not test positive. That's kind of what we would expect. And the Cornorhynchus townsendii, so the big-eared bats, also were not testing positive for PD either. So that's as we would have expected. If we move into load and attograms of PD, and when we see that, we can see that some individuals, so definitely the tricolored bats here, are ramping up to levels of PD loads that we do find concerning. So that suggests that they are going to be at risk in Texas as they have been other places. And Myotis velifer are showing higher levels in some caves in late February where it was not detected earlier in the year. So this is again cause for concern. They're not as high as the levels of the Perimyota subflavus, but cause for concern. Moving into central Texas, we see that there is one cave where we are not seeing any prevalence of PD in the Perimyota subflavus, but other locations where that is starting to ramp up, we should watch for that. And then on the far left here, we have a site where cave myotis are roosting with Tiderida brasiliensis, the Mexican free-tailed bat, and they have quite high levels of prevalence that ramped up between December and February. So that is concerning. And then when we look at the attograms of PD, we can see that the Myotis spellifer that are co-roosting with the Tiderida have very high levels of fungal load. Whereas if we just switch back to the fungal load that we were seeing in that same species in the North Texas caves, they were substantially lower. All right, so this suggests that Tiderida is going to be of less concern, but still something to keep an eye on and to look at this roosting situation. All right, so now I'm going to summarize what these findings kind of mean and then leave a bunch of time for questions. So it looks like tricolored bats are showing the same pattern of mortality that we've seen in other parts of their range. So even though I haven't known of any reports of tricolored bats dying yet, it seems like they are going to be at risk of white nose in Texas, as they have been in other parts of their range. Cave myotis do seem to be susceptible to white nose syndrome mortality, and so our swabbing is reflecting what we are now starting to see in terms of a report of a dead bat confirmed with PD and other suspected cases. And then co-roosting with free-tailed bats may increase fungal loads in this species. We found that in one roost, so that is worth investigating if that is a co-roosting situation or something else about that artificial roost. In terms of big brown bats, not pictured, and Townsend's big-eared bats here, as we've seen in other parts of their range, they do not appear to be highly vulnerable to white nose syndrome in Texas, so that's some good news. And so now, where do we go from here? So we have some idea of what's going on. So we're monitoring the progress of PD spread to, in part, understand which species are going to be susceptible and that we need to keep a close eye on their numbers and to see if we're going to detect the same levels of population decline, but also knowing which caves are having the invasion happen most rapidly means that they could be targeted for some form of cleaning or treatment that is still in the works or being developed or will be developed. So a number of cleaning and treatment trials have been done over several previous years at several different sites. There's nothing that we have as a magic bullet that's going to fix that, but knowing where to go for those things is one of the first steps. And then another possibility is enhancement of fall foraging. So that's another project we're working on in other parts of these bats range, so farther north, to see if we can help bats fatten up more prior to winter so that they will still have the disease but be able to weather it if they have better reserves. If they enter in better condition, fatter bats tend to survive. So those are potential options. And with that, I have a kind of embarrassing picture of Nate in a Tyvek suit. I should have really featured that a little bit more prominently. But I want to thank all of you for your attention and all of our team who came and went caving and getting these samples. So yeah, I will then open things up to questions and flip back through slides to answer things.
Awesome, thank you. And as I said, sacred duty to show embarrassing photos in Tyvek. It's just, that's the way it is, and from odd angles and everything else. So Kristin, in the chat window, there were a couple of questions that I tried to answer, but I want to make sure that I did at least one of them right. Tiga wanted to know whether in those I forget, it's the figures you showed from Fred Frick's paper, the size of the circle, are the circles sites or are they individuals?
Here?
That one.
It's got to be sites if it's prevalence.
And then the size of the circle is proportional to sample size.
I believe so. Yeah, I would have to double check that paper.
Okay. Tina Jackson is curious whether the samples are taken in both December and February of the same winter.
Yeah, so this was all this previous December and February. So this is fresh off the press data. So just this past year.
Okay, and then I'm going to try to. Jonah's got an open-ended question, but we'll get to that one. I'm trying to understand what Robert was asking. Can you go back to maybe the beginning of your talk? There's probably maybe 3 or 4 slides in where you're showing population declines.
Yeah.
There we are. I think it's that one. So I think the question is about what is driving the population increases after there's a crash? Is it something to do with species, or is it something?
Yeah, that's a whole field of research, is like what's happening when we have these populations recover. So one possibility is you've had a huge selection event where basically there's this incredibly high natural selection pressure with a lot of mortality. And so there are some survivors persisting, and we're trying to figure out what it is about these survivors that lets them stay there. Having not collected this data myself, I'm not sure how much, like, yeah, and figures 1 and 2, that does seem to be a pretty steady increase. But
so one thing, if you don't mind me interjecting with my opinion, which I don't know if it's actually supported by data. So when I was up in New England, we did see a significant number of colonies of little brown bats, which are the most heavily affected species. They declined precipitously over time, and it was really dramatic. But a lot of the colonies that we were looking at, either the larger ones remained stable after they crashed or gradually began to increase. And there's a question as to whether that has to do with sort of a nucleating effect where bats are gravitating, like dispersed bats are gravitating to larger idealized roosts, or if we're actually seeing more recruitment and a sort of evolutionary rescue process going on. It's hard to say because it's been, I'm sorry, go ahead.
Oh, no, just to expand on that for the audience that might be less familiar, is so these bats, we don't know if they go back to the same cave every year, and chances are they don't, that they have this fission-fusion population dynamic where a bunch of them are in caves we don't even know about. So, and these bats, particularly Meadow species, they're roosting in these clusters for a functional reason. So they might be, yeah, like Nate said, we're seeing more immigration than necessarily population growth. Is that what you're getting at?
Yeah, that's what I was getting at. I mean, there could be positive signs in sort of the demographics of it all, or the population genetics of it all, but these changes are happening over the course of within 10 years, so it's really hard to reach out to an evolutionary answer when there could be movement ecology answers waiting for us.
Absolutely, and that's part of what gave me pause, was looking at the scale of what would be colony size growth, and because bats reproduce so slowly, having 1 pup a year, and are probably much less likely to have pups in years when they're recovering from white nose syndrome, that was part of my furrowed brow as to that rate of population increase.
Okay, so let's get to Jonah's open-ended question. Can you speak to ideas of why we've seen die-offs in Central Texas but not in the Panhandle? And for those of you who aren't familiar, or I'm pretty sure Kristin pointed it out, but there is, this year we've had a significant mortality event of Myotis velifer in those red counties. And in fact, today the national map has been updated to illustrate white nose syndrome in a number of the counties in Central Texas. However, the same species of bat down there as it is up in North Texas, and we're not seeing mortality up north, which doesn't make a lot of sense in the ways we think about it. So there you go. There's some background.
Yeah.
Well, could it be the humidity?
So we haven't analyzed humidity data in the differences in these caves yet, so there could be some microclimate differences. But so it's definitely possible that they're in different types of caves, but then you would expect the more humid caves to be the ones where you're seeing more mortality. Yeah. Okay. I'm gonna stop, pause, make my sentence. It might, I think possibly we're better at detecting them in Central Texas because we have more people there to notice. A lot of these northern caves are pretty remote, and when these bats leave hibernacula to die a lot of the times, so we noticed that in the northern states and in Canada because seeing a brown bat on snow is pretty obvious to see, but when they're dying in rural areas and a brown bat on sand is much less obvious. I'm wondering if we're just not detecting them because they're not necessarily dying in these caves, particularly when it's easier for them to leave the caves in search of food.
Kristin, this is Jonah. I'm going to ask a follow-up question if that's all right. So the interesting thing that's been sort of a head scratcher for us is that we detected PD in the Panhandle at least 2 years before we detected it in Central Texas, and then we haven't really seen much of the ramp up until this year. So last year we saw a big ramp up in PD loads and prevalence in Central Texas, whereas in the Panhandle it stayed flat, even though it's been there for several years longer. And so I would suspect that it's not a lack in detecting. I mean, certainly there could be a die-off happen, but the swab results suggest that really the white nose hasn't really taken root there as aggressively. And again, this is just an open-ended question that's there are no real answers to. And so I didn't mean to throw a wrench into your presentation, but it's just something that I've been thinking about, and maybe for members of the audience to think about as well. I mean, you can see the national map here. PD spread through North Texas down into Central Texas, and yet for some reason we had this really aggressive ramp up, and a lot of Myotis velifer die this year in Texas. Yet the same species up in the Panhandle so far seems okay. The ramp up we've had this year is concerning. Like I would predict based on that we'll probably have a die off next year, but it's still interesting.
No, I agree, and I think part of my brain is just trying to figure out, like, I think there probably are microclimate differences that are driving this. I just haven't decided what I think they are yet based on the data. Because there are several humidity changes, there's several different patterns in temperature. So these caves fluctuate in their temperature throughout the day and throughout the year and have different ramps in those temperatures. And so how long a cave is cold before it decreases or how warm it was when it started, it's not just the average temperature, it's kind of what that temperature profile is.
I think Sarah Fritz was asking a question related to that. What about colony size? Would that have an impact, you think?
I mean, when you have these larger aggregations and more movements, I think you have more potential for introduction of the pathogen or for the pathogen to establish in a host that's not doing as well, and then for that host to produce high levels of fungal load that it can then spread to the other individuals of the colony. So when these bats wake up during their arousals, they don't just stay in their little spot and sleep and fly around. They also crawl over other bats. They mate with other bats. They likely switch hibernacula. So some of that is probably related to the movement. And I think I'm going to draw another parallel here is when you have places that have a lot of influx and outflux. We look at cities like New York City and COVID, where you have a population that has a lot more flux, you have more chances that that will get introduced. Where you have more remote areas with smaller population sizes, maybe there's less chance that the pathogen will take hold, but once it gets there, it would have the same effect.
Okay, so let's move on to Tiga has a question here. It says, any support for the quote shorter winter hypothesis, quote unquote, based on increased rate of PD loads through time?
So right now we're seeing PD loads that are characteristic of some of the northern sites where we have seen mortality, so around 8 attograms of PD. So what that says to me is even though we have the shorter winter, that shorter winter isn't enough to offset for the other factors because the fungal loads are still reaching those concerning levels.
Okay. For the bats from Bird Mama, for the bats that saw a spike in February, what were the confounding factors that would contribute to that? So would we see a spike through just growth, through fungal growth, or is there something else that could be driving that?
So I think it's that combination of what time of winter they were infected, which it probably has something to do with the social dynamics over winter, and then compounded with microclimate. So if those bats are roosting in more humid areas where the fungus grows better, that could increase those higher loads.
Okay. Have there been any complete eradications in particular caves in Texas? As far as we know, this was the first. I'm sorry, if you don't mind, I'll jump on this one.
Please do, Nate.
We know this is the first time we've seen mortality due to white nose syndrome, and so far, no, there have been no totally lost colonies, but we're not sure. We're not even sure how many bats we started with, which is the problem with everywhere across the country. Don't know how many bats we've started with, so it's really hard to know how much populations have declined. As I said, we haven't had extirpations in certain sites, but it's this was the first year. There's going to be declines coming for the next couple of years, so stay tuned. There may be worse news coming. Okay. Has there been any further insight on cave or roost substrate differences between Panhandle and Central Texas? So I think we had a conversation on one of these big white nose calls about limestone down in the Edwards Plateau versus gypsum up in the North Texas caves, and if that could have an effect. I'm not sure. Can you say anything to that, Kristin? I haven't heard anything new about that.
I haven't heard anything, and seeing these levels that they were able to ramp up in the gypsum caves, I think our consensus is that it doesn't look like that's going to be a protective substrate.
Okay. What does the fungus need to grow or survive?
All right. Well, it seems to need a cool, moist habitat to grow. It looks like it's not necessarily growing in the substrate so much as in the bats and then being shed onto the substrate, where then they pick it up and become more heavily infected and then shed. So I think it's safe to say that it's not actively growing in the substrate to levels that seem to be driving disease dynamics, and part of that is because substrates have lots of other microbiota in there. And then it's not going to grow at the warm body temperatures that the bats are at during the summer. Do you have anything to add, Nate?
Huh?
Oh, do you have anything to add to that one?
No, I mean, yeah, it's a cold environment and something to eat. I think, let's see here. So this next one's sort of directed toward me. How would I get an info list for the PD updates concerning my counties for Bastrop and Caldwell County? The best way to do that, Robert, is to go, if you check on whitenosesyndrome.org, it's the site that's dedicated to the national response for white nose syndrome, and those county updates happen when those should happen yearly-ish when we're able to compile our data and upload them to the site. So that one is pretty much live updates once the biologists have their data together to put things. And that takes a lot of steps of waiting for results to come back from the testing facilities and which are in Arizona and Wisconsin. So it takes a little bit of time, but that's the best site, whitenosesyndrome.org, all one word. Are Raffinese big-eared affected by white nose? I see that Liberty County is marked on the map. The Liberty County mark is that we found the fungus there. But Raffies, I do not believe are affected by white nose.
I think they're much like the other big-eared bats, that they're not in microclimates that are as susceptible.
Okay. What are your thoughts on how it got introduced to North America and now in Texas? Was it migration?
Okay. Well, I think the consensus is that this was human transmitted inadvertently on likely someone's caving gear. That's the best hypothesis we have to date. So someone unwittingly would have done that. And then once it appeared in North America, bats seem to have done a very good job of spreading it themselves as they're highly mobile creatures. It seems to be more driven when they're having the higher fungal loads. And so same with when it got to Texas is you can see that I'll go to the color progression map of where this started, and then it kind of moves down these mountain range areas further and further and kind of gets daisy chained passed along over years.
And one fun aspect of an additional thought about that is that there have been lots of ideas as to how white-nosed got here. Either it came on somebody's caving gear. There's people think that, or people have suggested that a bat came over in a shipping container, a European bat came over in a shipping container, or a Chinese bat came over in a shipping container. Yeah. It could be that there's a hypothesis that somebody brought a bunch of French cave soil to make French cheese in a New York cave.
I hadn't heard that one.
I've heard somebody say that Monsanto developed it so they could increase their profit margin on pesticides because that provides such a good service for pest management.
Oh man,
ideas, but I think that the one that that Kristin was talking about, the one that I think most of us agree on, is that it was inadvertently brought here on equipment.
And I guess whitenosesyndrome.org has good decontamination protocol and things that we follow, which is why we're in the fun Tyvek suits.
Yeah. Is there any research on possible transfer to non-cave dwelling species that may intermingle? So are there other bats that can get it that aren't cave obligate?
Yeah, I don't think anyone has. I think we've seen a silver-haired bat test positive for PD, and I don't think we've seen disease. I think that happened out in Oregon. So I don't think we've seen that play out. And I think part of that is because those non-cave hibernating bats aren't spending their winter in the microclimates that PD likes to develop. But it certainly is something that's been on our minds. As someone who studied silver-haired bats for my PhD, I started to really wonder if they could become infected because every once in a while they will turn up in a hibernacula.
Okay. Can you describe what the lesions on wings and muzzle look like during an active infection? Is there active blood? So is there circulation happening at the same time?
I think this is really your specialty, Nate.
Yeah. I wasn't going to jump on it, but
please do.
Yeah. So this is what I sort of did my PhD on this work. The lesions are what are called cupping erosions, where the fungus is growing in these masses that start to digest down into the skin tissue and infiltrate into the dermal layer of the wings. Because these bats are hibernating at the time, they're very cold. Their physiology has been turned way down. They are not actively bleeding, but there are holes in the wing tissue. So that's where, as Kristin alluded to, that's where we think this dehydration hypothesis could be coming in, where if you destroy the waterproofing layer of a mammal's skin, they lose a lot of water to the environment. And we see this happening with burn victims, human burn victims. One of the things you have to be very careful of is that they don't become massively dehydrated. So to answer your question, no, they're not actively bleeding, but if they were more active, perhaps we would see that. It's hard to say. I mean, when they arouse, they don't bleed. So I hope that answers the question. Where are we now? Has anyone been monitoring bats in West Texas? We are doing, I think BCI actually has some sites out there, right?
Wait, okay, yeah, I was mixing up east and west in my brain. Yes, we're starting to monitor West Texas caves. So it's a matter of finding a lot of them is the tricky part, is finding ones. We're exploring caves right now, and or not presently, but I had been looking for caves and hadn't found overwintering populations of these species yet. But yeah, we're still looking for where they are in West Texas.
Yeah, that's one of the areas where we need more eyes out there, but it's very hard to work in that region and find animals. We do have a new wildlife diversity biologist out in West Texas, Krista Demery. She is a very good bat biologist, and so hopefully she'll be doing a lot of cool work out there with bats, in addition to everything else that that's on her plate. Just FYI, we're down to something like 10 minutes left before 1 o'clock, so we'll pull the plug at 1, but we can still keep going with questions here. Blah blah blah. Tiga wants to know: are you or somebody working with Mexican counterparts? Are you?
As far as I know, BCI doesn't have any white nose stuff going on in Mexico for this. We have collaborations with Mexican counterparts for other things. So I think as this becomes more concerning, that's something that's on our radar.
Jonah, are you aware of any of that? Our focus is meant to be for Texas, but we do have an international coordinator in our staff.
Yeah, we do, and I am not, and I think that some of what we've been seeing happen in this last year with the die-off in Central Texas, potentially has some worrisome ramifications for what could happen in other southerly or temperate latitudes, because typically we all thought that white nose syndrome was primarily going to have impacts up in the north and cold regions where bats hibernated for long periods of time. But it appears that might not be the case so much. And so that's a very astute thing to point out, and something that's been on our to-do list as far as things just to tap in on and see. I mean, I have no idea if people are swabbing or testing for PD or looking for white nose in Mexico. So I'm sure there are some, but I'm not sure about the border regions right near us.
Okay, I think we've answered this next question. Are there now rules about stringent cleaning of equipment? Yes, there's the white nose decontamination protocol is on whitenosesyndrome.org and updated fairly regularly by the feds. The next question is, okay, is the white nose decontamination protocol working for Texas in regards to those caves that state and federal organizations manage? Are we allowing folks from out of Texas to use their own repelling gear, or is there a process we or the grottos are using to loan out gear? Are we micromanaging the recreation access to our states and caves to mitigate the potential spread of white nose syndrome? Okay, well, so the white nose syndrome decontamination protocols have been in place for the entire country for, I don't know, probably close to 10 years now. And my opinion is that yes, they work for human movement of the pathogen to new places. The state and federal cave access is usually managed in a way that visitors are asked to dip their boots or not take equipment that they've had in a cave into one of our caves. There's no real, we can't actively enforce it. We can't tell people, no, you can't bring your camera. We ask them, hey, if you did have your camera in another cave, can you please not take it inside this cave? And it's hard to regulate because, yeah, I mean, the grottos are being very good about it, and we're working with them, and they're understanding about the concerns to bats. But there are rogue people, rogue cavers out there who won't listen, and that's hard to control. So there's methods in place to regulate this a bit, but it's very hard to enforce. The positive news,
Nate,
sorry.
Oh, this is Jonah. I was just going to chime in on one piece. I didn't mean to cut you off. I'll wait till you're.
Oh, okay. The positive news, if you can call it that, is that it looks like the movement of the pathogen is far more driven by the animals than it is by humans.
And I was just going to restate that the Texas Parks and Wildlife does not really have any management authority over caves on private lands. So we can't require through any kind of force of law, any regulations that prevent people from going into caves without using decontamination protocols. We definitely require them in all of our research permits. So anyone doing research has to do decontamination. And on any state-owned and federal-owned lands, they're also required. But on private lands, it's all voluntary.
Okay, where are we now? Where did white nose syndrome originate? I think we touched on that.
Yeah.
You know, Europe, Eurasia, and also New York. Okay, here's a good one. Is this fungus similar to a virus in that it becomes less virulent over time? And is anyone using bat detection dogs for detection of the disease?
I mean, the idea would be that like there are several European bats that seem to be persisting with PD and even showing fungus growth on their face, that they survive the winter and they have different overwintering ecology, and it seems like they've co-evolved for a long time. I'm not sure if we've done any infection trial things to tease apart the fungus in North America becoming less virulent. Nate, are you aware of that?
No, but it's, oh, there we go. Are we seeing, yeah, everyone's seeing that page now, right? Okay. Mm-hmm. No, but it's tough because there could be, we see
it's really hard to tease apart that part versus all the other things that are happening with like the species adapting and different climates and different regions.
Right. And this is all happening on a very short period, over a very short time frame, and with animals that have a very long generation time, have a low reproductive rate. So it's hard to say that we're seeing any real signals in the data. We could just be seeing long-lived bats live for a long time, and perhaps if these bats live for 30 years, perhaps, which they do, perhaps, you know, once those old female bats eventually die, then we may lose those remnant populations. That's me speculating wildly, but it's you know, could be possible. Okay, we got 2 minutes left. So has white nose syndrome? Oh, real quick, sorry. I'm sharing this slide now. If you want to sign up for more information from our webinars, here's the webpage you can go to. Jonah pasted this up in the top of the chat. Next month, we're going to be talking about strategies to mitigate the impact of wind energy on bats. So, you know, how can we prevent bats from being killed by wind turbines? Okay, I think this one will have to be our last question, and then I can, you know, folks are welcome to email us to ask more if you want to. So, has white nose been identified in bats roosting in abandoned mines, or do they not have the appropriate microclimate?
Abandoned mines are, yes, basically. In short, yes, abandoned mines can be a place where white nose syndrome occurs.
Yes, exactly. And I think the microclimate could affect the way white nose progresses, but the fun part about that is that the bats like to choose the spots that are ideal for them, which then in turn becomes the spots that are ideal for the fungus.
Yeah. Well, thank you for giving me this opportunity to share some of the work we're doing.